DNA is the genetic information in genes. It is transcribed into RNA, and every 3 base pairs of the RNA is codons. The codons are translated into amino acids. The amino acid chain makes up a protein, and the protein is what is responsible for the regulation of gene expression. When there's an alteration in the DNA, this causes a mutation. A point mutation is the substitution of a single DNA base pair for another. And there are 2 types of this substitution. We have transversion and transition. Transversion is a pyrimidine to a purine, or vice versa, or pyrimidine to pyrimidine, or purine to purine. And it results in 4 types of point mutations. These mutations can either result in the loss of the function, gain of function, or no change. Our original DNA sequence reads TGG. The G is mutated to an A, which is an example of a transition. Our new codon now reads UGA, which translates into stop. This stop amino acid prevents the rest of the RNA sequence from being translated, resulting in a shorter protein. Here our original DNA sequence reads TGC. The mutant strand reads TGT. The C to a T is an example of a transition. When this mutant strand is transcribed, its codon is UGU, which translates into the amino acid CYS. The original DNA non-mutant strand also translated to CYS. This no change in the amino acid is an example of a silent mutation, which results in a no change to. Here our original DNA reads AAC. The mutant strand changes the C to an A, which is a transversion, and now reads AAA. When it is transcribed into mRNA, the codon now reads AAA, which translates to the amino acid LYS. The original non-mutant DNA sequence translated to ANS. This single amino acid change is considered a missense mutation. In our final mutated DNA strand, we have a C rather than a T. The original T changing to a C is an example of a transition. Our new mRNA codon reads CGA and is translated into the amino acids ARG. Originally, the mutant DNA had that amino acid coding as a stop, but because it's no longer a stop and it continues to translate the next couple codons, this is considered a read-through, which results in a longer protein.